In 3.1.6, they want us to talk about how by changing one base, and that's what this means, the consequence, what happens when you change one base for another? What happens if you change a C for a T? What is the effect? And the example they're going to use is sickle cell anemia. And I know we've talked about this before, but we're going to relate it not only to like how the consequence is related to a different codon or how it's a different amino acid. We're going to look about how the consequence can lead to a change in the allele frequency. So we talked about alleles being the form of a gene, and the form can be a normal hemoglobin or a sickle cell hemoglobin. So those are two forms. And the consequence can also be evolution. So we're going to look at this as an example of human evolution. So let's just quickly talk about the base pair substitution, the effect in review, and then we're going to talk about what it physically does to a person who has sickle cell. So we've talked about already that if we're looking at 1, 2, 3, 4, 5, the sixth triplet of bases in the hemoglobin gene. Hemoglobin has 4 parts to it. This is one of the beta subunits. We don't really need to worry about that now. But the sixth one, the normal amino acid with that is glutamic acid. So that's what happens when the gene is normal. However, if we've changed one letter, if we change that T to an A, we get a different codon on our messenger RNA, right? This is transcription, and then messenger RNA to protein is translation. By changing that one letter, now our codon is GUA, and we get a different amino acid in location 6 on this beta subunit of the hemoglobin protein. So what? Like, how does this affect anything? So let's just move to the next slide. And you can see it. I think I stole this from Cognitive, but they did a very nice job of explaining this. So in a normal situation, your beta subunit is like this, right? And if we're looking at it from the top, you have 2 alpha subunits, 2 beta subunits. And by having this spacing out like this, it's very easy for it to have oxygen associate with it. It will easily connect and carry it around. So it's a very effective protein for doing its job of carrying oxygen, right? That's what hemoglobin does. Hemoglobin's job is to carry oxygen around to all the different parts of your body. However, by changing just that one amino acid to valine, we get a different shape. It has to do with its hydrophobic or hydrophilic properties. By having this little shape here, what happens is it allows another beta to fit in there. I don't know if you can see that down here. So here's a beta, 2 alphas, and another beta. And what happens is it forms not like a beautiful spaced out shape like here, where they're all kind of individual. It makes them join together in this very long fibrous shape. And 2 things happen when it's like this. One, it actually pokes holes in your red blood cell and kind of has it leak out, so it gets this weird kind of shape. The second thing it does is it has a very stiff quality to it, so it's not flexible. The beauty of your red blood cells, when they're like this, is that they're squishy and they can squeeze to all different parts of your body, but when they're like this, they get stuck in your blood vessels and they cause something called a sickling event or a sickling crisis. And it can be very painful. It can lead to tissue damage and even sometimes death.